Hello, Bio30, and welcome to the video on the Synapse Lesson 4. Today in this lesson, you are going to learn what exactly is a synapse, what and how neurotransmitters are used in the movement of an impulse from one neuron to another, and then how essentially the impulse is sent across the synaptic cleft and moving from the axon endings to a new set of dendrites. So we're going to start off by defining what is a synapse. We have looked at a neuron, and we have looked at it as one single entity from dendrites to axon terminals or axon endings. And the reason is that we look at one is because neurons don't actually touch. There's a little bit of space between the axon terminal or the axon endings to the dendrites. So what has to happen is info needs to jump from one neuron across a space called the synaptic cleft and move to the next. So if you see this picture right here, this would be the end of one neuron and the starting of another. So to define synapse is the space between two neurons. It's approximately 20 nanometers in length, in size, and the transmission across a synapse is very slow. There are many synapses that occur between two neurons, and an impulse moves from presynaptic to postsynaptic neuron. Now, those are two really key words that you're going to need to know because we're going to use them a lot as we discuss. So presynaptic and postsynaptic. The nerve endings of the presynaptic neuron are filled with synaptic vesicles and contain neurotransmitters. So what this is essentially saying is axon endings are essentially what we call presynaptic spaces. So the axon endings are presynaptic, and they're filled with these vesicles that can contain something called neurotransmitters. Now, it's important to understand what a neurotransmitter is because it's found in lots of places, not just across the synapse. So essentially, neurotransmitters are chemicals that, in this case, move the signals across the synapse, and they're found inside synaptic vesicles. But a neurotransmitter can be found in lots of different areas, especially in your brain. It can attach to little channels on your brain or little receptors, and that's things like dopamine and serotonin. Maybe you've heard of those. And essentially, neurotransmitters, help us with different feelings in the brain, and in this case, for us, helping the neuron pass on an impulse. Now, the word exocytose or exocytosis means to burst open. Okay, exo meaning exit, and then essentially, I believe the root of exocytosis or exocytosis. The tosis part is to break apart. And what happens is when those neurotransmitters break from the synaptic vesicle, they diffuse across the synapse and attach to ion channels on the dendrites of the next neuron. We call these excitatory neurotransmitters, and they attach to the sodium channels on the other end, which are the dendrites. This is really key because excitatory neurotransmitter means that it'll open up channels. It excites the channels and opens up the channels. So let's look at this in a little bit more detail. You have a picture to draw the sequence that I'm going to go through. You may want to go back and stop and redo it at the end, but we'll see how this goes. All right, so here I got a couple neurons. Again, not touching you, how your siblings said that. This is exactly this moment. Okay, so here we go. We have ending of the axon. So the first neuron, we call this the presynaptic knob, and it's filled with tons of vesicles with neurotransmitters in them. And our neurotransmitter for this case, and this is important, you need to know this word, is acetylcholine. Here we have the postsynaptic knob, and in between we have the synaptic cleft. Now the postsynaptic knob is the dendrite of the next neuron. So the first step that's going to occur, and you can follow along and add these into your notes, you have the steps as synaptic activities, is the nerve impulse goes down the presynaptic axon. Sodium is rushing inside of the axon membrane, and we then in this process trigger calcium. So calcium ions signal a synaptic vesicle to start moving towards the presynaptic knob. So here we've now triggered calcium to come in and push these vesicles all the way to the edge of the presynaptic knob. What ends up happening is the synaptic vesicle merges with the membrane of the presynaptic uh, knob. And what happens is it through the process of exocytosis, the synaptic vesicle releases acetylcholine into the synapse or into the synaptic cleft space. So that's what those are right there. Now, acetylcholine will diffuse across our synaptic cleft until it reaches the postsynaptic knob. That's on the other side, which you can see right here. Now, on the postsynaptic knob or on these dendrites, right, is once acetylcholine gets there, it's going to attach to sodium channels. So you can see there's a little space for it to park itself and attach itself to the sodium channels. Will eventually make their way in. And what it does is it opens up those channels to allow sodium to pass through. We call this depolarization. We know this from the neurotransmission video, that as soon as sodium comes inside of any axon or any passes through a membrane, we are going to get depolarization or an action potential. And that's the same process here. So once the channels are open, the sodium will begin to flood through into the postsynaptic knob, and it'll travel down the dendrite through the cell body into the axon, and then again begin that wave of depolarization down the postsynaptic axon, which is essentially just the neurotransmission. Now, this is really key here. We don't want to leave these sodium channels open, because if we left these sodium channels open, these neurons would constantly try to go through a wave of depolarization. They would constantly try to say that there's an impulse being sent. So we need to close them. So in order to do this, we have to remove acetylcholine from the situation. Acetylcholine is what's holding open the channels. We want to close them. So in comes an enzyme called cholinesterase. Now, cholinesterase and acetylcholine, you know they match because if you look at the end word of acetylcholine, choline, and then look at cholinesterase or cholinesterase, you'll notice that they match. So cholinesterase is the enzyme that specifically breaks down acetylcholine. Because remember, enzymes have specific substrates that they attach to. And we know what they attach to because it's usually the name of the substrate and then ASE on the end, which is what cholinesterase is. So it comes in and it breaks off acetylcholine from the sodium channels, and it allows the sodium channels to close. So in comes cholinesterase, removes acetylcholine, channels close. Acetylcholine is then eventually released from cholinesterase because not all enzymes break down and break apart break apart their substrate. Instead, what they'll do is they'll release the acetylcholine. So acetylcholine is released from the cholinesterase, and it's actually reabsorbed back into the synaptic vesicles back where it started from to be used again. And cholinesterase goes back and gets reabsorbed where it came from to be used for later times as well. So in this lesson, you have learned what is a synapse, what is a neurotransmitter, and how the impulse is sent across the synaptic vesicle. I would go back and make sure that you have all of the little bits and pieces filled in for synaptic activities 1 through 7, but also making sure that you've drawn the sequence as we've gone through it. If you have any questions, please let me know.